Okay, so welcome back to this next video in which we are discussing drug addiction. Okay, right, so we've just discussed the brain's reward system, okay, and we've discussed how drugs of addiction are going to hijack the brain's reward system. So let me just remind you of this because it's so important that we should go over it again. Okay, and I also would like to just add a few more little bits regarding drug addiction, okay, before we actually go on to looking at examples of addictive drugs, and the ones we're going to begin with are the psychostimulants, so cocaine and amphetamines. Okay, right, so remember, this reward system is all about teaching the brain, okay? It's all about causing remodeling of the brain so that pro-survival stimuli become wanted and craved, okay? And also so that you can form associations between the cues which tell you that you can get the stimulus and then the actual behavior that then will actually get you that stimulus. Okay, right, so the example that we are using is eating chocolate for the first time, okay, and I want to stress that this is not just someone who has eaten a huge number of bars of chocolate in their life eating another bar of chocolate. We'll come to that, okay, that doesn't require teaching anymore. It's someone who has never been exposed to chocolate before in their life, okay, being exposed to it for the first time, okay. They do not know what to expect. The brain does not know what to expect, okay? So this is an unexpected novel stimulus, okay? And what happens is that it actually turns out to be extremely good. It's very pro-survival, okay? The brain receives all sorts of information about how good this chocolate is. It also knows that it has no previous knowledge of this stimulus, okay, so it then decides that we need to now learn, okay, we need to remodel the brain. We need to remodel the brain so that the behavior of seeking this chocolate becomes more paramount, okay, so that we forge new memories and we remodel the brain so that we can get more of this stimulus, okay, which is going to aid our survival. Okay, so we then take the decision to activate the ventral tegmental area, okay, it then fires, okay, so loads of the neurons in there are going to increase the rate at which they fire action potentials, okay, and that means you're going to get an increase in dopaminergic transmission, an increase in the amount of dopamine being released into the nucleus accumbens and medial prefrontal cortex, okay, and this increase in dopaminergic neurotransmission in these two areas is believed to then underlie the remodeling of the brain, okay, so it triggers the remodeling of the brain, and then this remodeling of the brain causes you to crave and want the chocolate, the stimulus, okay, and also is going to cause these associations between the cues that tell you that you can get the stimulus and then the behavior that will actually get the stimulus from those cues. Okay, and I want to stress that dopamine itself is not responsible for the cravings and the wantings, okay? Dopamine causes the remodeling of the brain that then produces the cravings and the wantings, okay? And the idea behind these things is that it's going to lead to getting more of this pro-survival stimulus, okay? However, once you have learnt enough, once you've taken this stimulus enough times, okay, so once you've eaten chocolate enough times, okay, the brain knows now that this stuff is fantastic, and we do not need to establish more cravings and more wantings, and we do not need to establish more of these associations, okay, because it's completely expected. So now, this time, what will happen is the brain will still receive all the information about how brilliant the chocolate is, okay, but it will say, we know, okay, so we don't need to activate the ventral tegmental area anymore, we don't need to raise the dopamine in the nucleus accumbens and the medial prefrontal cortex, we don't need any further remodeling, and therefore we're not going to make our cravings and wantings stronger, we're not going to make these associations stronger. Okay, what happens with addictive drugs is that the addictive drugs bypass all of this bit up here. Okay, they just result in increased dopamine in the nucleus accumbens and the medial prefrontal cortex directly, and we're going to see many different mechanisms by which different addictive drugs work. Okay, so they cause the dopamine to go up. So it's not really true to say that they all stimulate the ventral tegmental area. Many of them do. Some of them are actually going to act on the axon terminals of the ventral tegmental area. For instance, the psychostimulants that we're about to look at are going to work on the axon terminals. What is true is that they all, through very crude means, very crude direct means, bypassing the entire brain, okay, they all through these crude means that we understand in all but the case of ethanol, okay, raise dopamine in the nucleus accumbens and the medial prefrontal cortex, okay, and what that then does is it means that you've continually got the teaching signal on. This continually says, now remodel the brain so that we get cravings and wantings for this addictive drug, okay, and so that we get associations formed between the cues which tell us that we can get the drug, so potentially drug paraphernalia, okay, people and places, okay, and then the actual behavior of taking the drug, okay, and this teaching never stops. Remember, the teaching for the chocolate stopped, okay, this never stops. Every time you take the drug, it's going to teach it stronger, it's going to make the addiction stronger. Okay, and then you get cravings and wantings that are out of control, basically, to the point where it becomes your obsession. Okay, it's what your life is about, just getting the drug. Okay, and you also get so strong associations between cues and behavior that they are believed to potentially start displacing voluntary control of behavior, so that when you see the cues associated with taking the drug, you just take the drug without voluntary control over that. Okay, it just happens, the brain just does it. Okay, so that's what underlies addiction, which is the continual taking of the drug despite the negative consequences. Okay, now the additional little things that I want to talk about are the concept of psychological dependence and psychological withdrawal. Okay, so the idea of psychological dependence then, basically psychological dependence means that you get into the state where you cannot function normally psychologically without the drug anymore. So this remodeling is so extreme, okay, if this remodeling continues and continues, it becomes so extreme that you develop psychological dependence, where you cannot function normally psychologically anymore without the continued administration of the drug. Now this is extremely linked to the concept of psychological withdrawal, okay, which basically means that if you stop taking the drug, bad things start happening. Your psychological state becomes subnormal. Okay, so what happens generally if you take, if you stop taking the drug, then the two main symptoms of psychological withdrawal are dysphoria. Okay, so you end up getting a terrible, terrible mood. Okay, that's what dysphoria means. It means a horrible, horrible mood. Okay, and you get intense cravings. Okay, which we can explain with the remodeling that has occurred to the brain because of taking the drugs. Okay, so what you must understand is that these addictive drugs, they permanently change your brain. They are remodeling your brain. Having permanently high dopamine levels induced by the addictive drugs whenever you take them is remodeling your brain. Okay, and this is what produces psychological dependence, and why when you stop taking them you develop psychological withdrawal. Okay, now there's a related concept, which is the concept of physiological dependence and physiological withdrawal. Now, it is not necessary at all that addictive drugs need to produce this symptom of physiological dependence and physiological withdrawal. Okay, some drugs do. The notable examples would be opiates. Okay, but you don't need to. There are drugs that don't produce it and are still addictive. Okay, so physiological dependence, and I haven't written this out, but physiological and then the word dependence after it would mean that your physiological systems can no longer function properly without the drug. Okay, and physiological withdrawal then means that if you come off the drug, you get physiological side effects. Okay, your physiology no longer functions properly. Okay, so opiates are the key ones that we're going to talk about, which cause physiological withdrawal. Okay, but this is totally different to their psychological effects. Okay, their psychological effects is because they're affecting this pathway here. Their physiological effects is because they're affecting loads of pathways in the peripheral physiological systems. Okay, so for instance, opiates famously cause cramping and diarrhea when you come off them. Okay, and that's because your intestinal systems, your intestinal physiology has grown used to the presence of the opiates, okay, and then when you suddenly remove them, it no longer functions properly, okay, it can't function without them. So you are physiologically dependent on the presence of the opiates. Okay, right, but we'll come on to the physiological dependence that occurs with opiate administration later on. Okay, we'll do opiates last because we'll discuss the physiological effects as well. The psychological dependence and the psychological withdrawal are related to the actual interference with this reward pathway here. Okay, right, so now what we're going to do is we're going to start our study of individual examples of addictive drugs. Okay, and the ones that we're going to start off with is the psychostimulants. Okay, and the aim of this discussion is to see how all of these different types of addictive drugs are going to raise dopaminergic transmission in the nucleus accumbens and medial prefrontal cortex. Okay, so we're going to then start off with the psychostimulants. Okay, so firstly, what are the psychostimulants? So the main two are cocaine, okay, and also the amphetamines. Okay, now there are multiple drugs that are classified as amphetamines, and we'll talk about 3 different amphetamines in a moment. Okay, right, so to understand the mechanism of these psychostimulants, we need to study the dopaminergic neurons in more detail. Okay, specifically we need to be looking at the axon terminals of the dopaminergic neurons, or if it's an en passant synapse, the synaptic boutons. Okay, but we'll imagine that it's a neural synapse and therefore that we've got an axon terminal. The principle is exactly the same in the case of a synaptic bouton. Okay, the picture would just be different. Okay, so let's say that this is an axon terminal of one of these dopaminergic neurons. So remember, the dopaminergic neuron has its cell body in the ventral tegmental area. Its axon is then being sent through the medial forebrain bundle, and then finally you're going into the nucleus accumbens or the medial prefrontal cortex, where you will have an axon terminal or potentially synaptic boutons which will be releasing dopamine, okay, onto another neuron, or if it's varicosity, just into the extracellular fluid. Okay, so we want to study this axon terminal here because this is where the psychostimulants are all going to act. They are going to act on the axon terminals or the synaptic boutons of dopaminergic neurons directly, and they're going to trigger either an increase in the release of dopamine from them, in the case of amphetamines, or they're going to block the reuptake in the case of cocaine. So let's study this in a little bit more detail. Okay, so firstly, let's just mention the synthesis of dopamine, okay, which is occurring in the axon terminal. Okay, now I'm not going to draw out the molecular pathways here. All we need to know is that you start off with an amino acid tyrosine, okay, so tyrosine is one of the proteinergic amino acids. It's used in proteins, so you know it's pretty essential for life. Okay, so you have it all over the place. So here is tyrosine, and you're going to go through a synthesis pathway to turn tyrosine into dopamine. Okay, so in the axon terminal, you're going to be getting the synthesis of dopamine from the amino acid tyrosine. Okay, now, before dopamine is actually released from the axon terminal, what happens is it gets stored in vesicles, known as synaptic vesicles. Okay, so you're going to put loads of dopamine molecules into a synaptic vesicle, and then when an action potential propagates down the axon, what will happen is these synaptic vesicles will be released. Okay, so you don't just produce dopamine and release it, instead you want to store it until an action potential comes along. Okay, so here, this is a synaptic vesicle here. Okay, right, So, the first thing that I'm going to mention is how do we actually get the dopamine from the cytoplasm of the axon terminal into the lumen of this synaptic vesicle. Okay, and this synaptic vesicle is a membrane-bound structure. Okay, so it's a separate compartment from the cytoplasm. So we need to get the dopamine across this membrane here. Okay, well this involves a secondary active transport process. So firstly, what I'm going to tell you about is the acidification of vesicles. So at first, this will seem a little bit unmotivated. It will seem a little bit off course. Okay, but we'll see how it's all going to work to get dopamine through. So before telling you about how you're going to get dopamine through, what I'm going to tell you about is something known as the vesicular ATPase. Okay, so this is for short called the VATPase. Okay, and this stands for the vesicular ATPase. And what this is it's a protein that is sitting in the membrane, okay, of the synaptic vesicle. So here it is in pink, shown as a ball here. Okay, and what this protein is going to do is it's going to be a primary active transporter. Okay, so what's meant by a primary active transporter? Well, primary active transporter is something which uses the hydrolysis of ATP or some other chemical reaction, but it's usually the hydrolysis of ATP, to provide the energy needed to pump a molecule up its electrochemical gradient. Okay, so we're going to be pumping protons, okay, the nucleus of a hydrogen atom, so H+, from the cytoplasm into the synaptic vesicle. Okay, now this means that you're going to end up with having a really high concentration of protons in the lumen of the synaptic vesicle, which means that the lumen of the synaptic vesicle ends up extremely acidic, because remember, the more protons you have, the more acidic you are. That's what pH is all about. It means how concentrated are protons. Okay, so you're going to end up with a huge number of protons stored within this synaptic vesicle. Okay, now, in order to continue moving protons in, once you've got a really high concentration in here, you need to expend energy. You need energy to do that. Okay, and the energy for this is provided from a chemical reaction. It's provided by the hydrolysis of ATP. So you're going to turn ATP, adenosine triphosphate, into an ADP molecule, adenosine diphosphate, and also an inorganic phosphate here. Okay, so here is the hydrolysis of ATP. So that is why this is a primary active transporter, because it is coupling the transport of protons into the synaptic vesicle to a chemical reaction, to the hydrolysis of ATP. And I'm sorry it suggests the phosphate is outside of the cell. That should be inside the cell. It was just, put there for space reasons. Okay, right. Then what you end up with is a really high proton concentration in the lumen of the synaptic vesicle, and we are now going to use this really high proton concentration in the synaptic vesicle to move dopamine into the synaptic vesicle. Okay, so the transporter that's going to transport dopamine is going to be what's known as a secondary active transporter. Okay, which means that you're going to transport dopamine into the synaptic vesicle, okay, against its electrochemical gradient, okay, which means that you're going to need energy, but you're not going to get your energy from the hydrolysis of ATP, not from some chemical reaction, basically. Instead, you are going to get it from an already established ionic gradient, okay, and in this case, we've got this proton gradient across the cell membrane. So the idea is this: if we let the protons leave the synaptic vesicle and go back into the cytoplasm, where they want to go, because that's down their electrochemical gradient, they would give out energy. We could use that energy that they give out to move the dopamine in. Okay, and that's exactly what we're going to do. Okay, so we are going to have a transporter here, okay, on the membrane of the synaptic vesicle, which I'm colouring in red here, and this transporter is known as the VMAT2. Okay, and the VMAT2 stands for, I'll put it down here, it stands for the vesicular, that's the V, and then the MA is for monoamine, okay, so M for mono, A for amine, and then the T is for transporter, so it's the vesicular monoamine transporter 2. 2. Now there is a vesicular monoamine transporter 1, but it's not generally found in axon terminals like this. Okay, the one that you generally find in axon terminals is the vesicular monoamine transporter 2. Okay, or VMAT2 for short. Okay, now VMAT2 is going to move one dopamine molecule into the lumen of the synaptic vesicle. Oh, and by the way, what's the origin of its name? Vesicular is obvious, it's in the vesicles. Monoamine means that it's going to be moving monoamine neurotransmitters. Remember, dopamine is a monoamine. Okay, so this transporter will move dopamine. It will also move the other monoamines, such as serotonin, noradrenaline, adrenaline, if they're present. Okay, but this is a dopaminergic neuron, so it's going to be moving dopamine, not those other ones. Okay, right, so it's going to move one dopamine molecule into the lumen of the synaptic vesicle in exchange for 2 protons leaving the synaptic vesicle. Okay, so you have to let 2 protons out for every dopamine molecule that's going to come in, basically. Okay, right, and because it's using the ionic gradient across this cell membrane to drive the movement of dopamine in, sorry, not cell membrane, this vesicular membrane, to drive the movement of dopamine in, it's not a primary active transporter, but instead it is a secondary active transporter. So it's really secondary to the establishing of this proton gradient, which was done by the vesicular ATPase here. Okay, so that's how you can store dopamine in these synaptic vesicles here. Okay, and then when an action potential arrives in this axon terminal, what will then happen is that will drive the fusion of these synaptic vesicles with the presynaptic membrane here. So this membrane of the axon terminal, this is known as the presynaptic membrane. Okay, so you're going to fuse the synaptic vesicle with the presynaptic membrane, and then what you'll get is the release of all of the contents of the synaptic vesicle into the extracellular fluid, okay, and that will then go off and act on receptors on the postsynaptic cell. Okay, now, the axon terminal then has a way of terminating the signal, because you don't want the dopamine released and then remaining there forevermore. You just build up more and more and more dopamine, okay, and that will be useless. Okay, you have to get rid of that dopamine once you have released it, okay. So there is a way of doing this, okay. The presynaptic membrane has on it a transporter which is capable of moving dopamine back into the cytoplasm of the axon terminal. Okay, and I'll colour this transporter in turquoise, and we need to know all of this, okay, because these are going to be the targets of these drugs. Okay, so this transporter here, this is known as the dopamine transporter, okay, or for short it's abbreviated as DAT. So DA is for dopamine, okay, and then the T is for transporter. Okay, right. So what then does the dopamine transporter do? Well, the dopamine transporter is going to, again, be a secondary active transporter. So it is not going to couple the movement of dopamine to a chemical reaction. Okay, so where instead is it going to get its energy from? Well, it's going to get the energy to move the dopamine into the axon terminal through allowing ions to move down their electrochemical gradient. So you will know that sodium ions are much higher in concentration extracellularly than they are inside the cell. So usually it's around a concentration of 145 millimolar extracellularly and around a 12 millimolar concentration of sodium intracellularly. So that's around a 12-fold gradient of sodium ions. Okay, chloride anions, okay, also generally have a concentration gradient favouring the movement into the cytoplasm of the axon terminal. I don't know the figures off my head, but chloride concentration is usually higher extracellularly than it is intracellularly. Okay, you are going to allow 2 sodium ions and 1 chloride anion to come into the cell, okay, like so. So I'll have to put 3. So here we have 2 sodium ions coming in, okay, and 1 chloride anion there, okay. Those are all going to come in exchange, well, not in exchange, so long as one dopamine can also come in. Okay, so this one was an exchanger. This one, they're all moving in the same direction, so that's known as a symporter. Okay, so when you put things, when all the things that are being moved together are going in the same direction, it's called a symporter. Okay, so you're getting these two sodium ions in yellow there, and you're getting one chloride anion in pink here. And that's so that a single dopamine molecule can come back into the cytoplasm of the cell. Okay, so here is that dopamine molecule. Now, the sodium ions, when they come in, they will release a lot of energy. Okay, not so much for the chloride anion, and you might be wondering, well, why? It's going down its concentration gradient, but you also have to factor in the electrical gradient across the cell membrane. Okay, and generally the electrical gradient across the cell membrane will be something around -65 millivolts. Okay, which means that the electrical potential inside the cell is 65 millivolts lower than the electrical potential outside the cell. Sodium ions, they want to be where the electrical potential is lower, so they want to go into the cell. Okay, they want to go down the electrical gradient. Chloride anions, being negative, they do not want to go where the electrical potential is lower. Okay, so they're not going to want to come into the cell. So because of that, even though the concentration gradient would say, yes, when you bring chloride in, you'll release energy, the electrical gradient is kind of going to balance that out. Okay, so really this isn't going to release that much energy at all. It might even cost you a bit of energy. I don't know what the exact numbers on that are. Okay, but the two sodium ions coming in are definitely going to release energy, okay, because they're going down their concentration gradient, they're going down the electrical gradient, so both of these factors mean that you're going to get the release of energy, okay, and that's what's going to drive the movement of dopamine in. Okay, right, so that's how we're going to reuptake dopamine into the axon terminal. Okay, so all of this was necessary to now understand how the psychostimulants are going to work. Okay, so let's start off with cocaine. Okay, so how does cocaine work? How does it produce addiction, is what we're asking. We're not asking how does cocaine produce all the other effects it produces, we're asking how does it produce addiction. Okay, so this is not the explanation for all of the effects of cocaine, this is the explanation for its addictive effects. Okay, so basically cocaine binds to and blocks the action of the dopamine transporter. Okay, so let's draw this. It's going to bind to the dopamine transporter here, and it's going to stop that from working. Okay, so that you can't reuptake dopamine anymore. Okay, now you will remember me telling you that there is a basal level of activity in the ventral tegmental area neurons. Okay, they're continuously firing at a low level and releasing some dopamine into the nucleus accumbens and the medial prefrontal cortex. Now, that dopamine will be usually being reuptaken by the dopamine transporter, okay, and therefore will produce a basal signal which will not produce the changes that remodel the brain to produce cravings and wantings and memory associations. Okay. However, if we now block that transporter, we stop the uptake of dopamine back into these dopaminergic neurons. Okay. So the dopamine is going to remain in the nucleus accumbens and the medial prefrontal cortex, and you're going to get much more effect of that dopamine. Okay. So dopamine levels are going to now remain high in the synaptic clefts, okay, and therefore the amount of stimulation that you're going to get of dopamine receptors is going to go up hugely, okay, and that then is going to do the same thing as you would get if the if the ventral tegmental area had actually increased its firing rate, okay, and that then is going to produce remodeling of the brain that's going to produce cravings and wantings and memory associations. Okay, right, so that then is how cocaine produces an increase in dopaminergic transmission in the nucleus accumbens and the medial prefrontal cortex. Okay, right, and I think we'll call it there for this video, and in the next video what we'll do is we'll look at firstly the 3 different examples of amphetamines that I'm going to talk about, and then we'll see the common mechanism of action by which they all work to cause the release of dopamine from dopaminergic neurons.